Welcome back to Water Quality in the Pacific Northwest. We're currently covering a series of lectures that deal with surface water quality. We're looking at different pollutants and we're looking at the measurement of these pollutants. If we take a look at the components of this unit, you can see that we've covered nutrients, sediments, erosion on land. We've had 2 lectures on the process of eutrophication. We talked about temperature. And today we're starting the first of 2 lectures on macroinvertebrates. So our lecture today, lecture number 16, is macroinvertebrates. Let's take a look by starting at a background to what are macroinvertebrates. Macroinvertebrates are important because they're commonly used as a biological assessment to evaluate the biological integrity of small streams. This is the most common measurement we use to determine water quality in small stream segments. These biological assessments attempt to quantify complex ecological processes into a single score, or what we call an ecological health rating. There are advantages of biological assessments over traditional chemical assessments. These include things like, first of all, biological assessments can be less expensive, and they can detect the effect of pollutants on the environment. And we can do this by coming up with a composite single score. There are many different types of biological assessments as a background. Biological assessments of streams have been developed for using algae as an indicator, using bacteria as an indicator, using fish as an indicator, and then finally, the topic of today's lecture, using benthic macroinvertebrates as an indicator of the quality of the water in the stream. Now, simply defined, benthic macroinvertebrates are aquatic organisms that are found in the bottom of the substratum of the water bodies. These are organisms, aquatic organisms, that live in the bottom of the substrate. They're attached to the cobbles in the bottom of streams. Benthic macroinvertebrate populations are the most commonly used in the community. They're the most commonly used community in biological assessments of streams because they have some very important attributes. One, macroinvertebrates indicate localized conditions within the stream because they are relatively sedentary. They don't migrate up and down the stream. So what you find in a particular area of the stream is very indicative of the actual water quality in the stream. In addition, macroinvertebrates indicate integrated short-term environmental impacts. Short-term environmental impacts because they have very short life cycles. So you know what's going on now. Another attribute which makes benthic microinvertebrates an important indicator is they allow experienced biologists to rapidly and easily examine water quality conditions. Biologists go out and take samples, and a good biologist can identify the different species of macroinvertebrates that are in their samples. Another important aspect that make macroinvertebrates a preferred biological indicator is that macroinvertebrates possess a wide range of trophic levels and pollution tolerances that allow for comparison. Certain macroinvertebrates are not tolerant to pollutants. So if you go in and take a sample and that macroinvertebrate is not present in your sample, the odds are a particular pollutant is actually causing the problem in the stream. In addition to that, macroinvertebrates are food. They provide a primary food source for fish and other types of aquatic organisms. And then finally, macroinvertebrates are relatively easy to sample. There's a standard procedure. You'll see that procedure in tomorrow's lecture. They're easy to sample, and they're pretty economical to sample compared to going out and taking samples and doing chemical analysis. They're relatively economical to do. Now, with all biological indicators, there are a few drawbacks. The drawbacks, or the primary drawbacks that I think that are important, are that spatial and temporal variability is inherent. Each sampling point within a stream has its own specific habitat structure, which means you can take a sample at point X in the stream and just walk 2 or 3 meters upstream, and you'll see big differences within a few meters. Thus, we have to have a good sampling regime. We just can't take one sample in a spot in a stream. We have to take multiple samples and average those samples to get a good indication of the biological integrity and thus the water quality in the stream. So sampling, we have to account for that variability when we sample. And then the second major drawback is that if we have unexpected storms or we have major runoff events, they can cause declines in localized populations that may not represent the watershed as a whole. So we have to take that into account when we actually look and analyze the data. But overall, despite these two mentioned drawbacks, macroinvertebrates are some of the best biological indicators available for water quality analysis. And thus, it is relatively commonly done. There are lots of consultants out there that regularly make their living by doing macroinvertebrate sampling and analysis and making recommendations on that. What kind of programs do we have to assess streams using macroinvertebrates? We have what's called rapid bioassessment. Rapid biological assessment protocols, commonly use the acronym RBP, gather data to make management decisions rapidly. You can gather data fast and make management decisions in a relatively short period of time. RBPs are used to predict conditions relative to what's considered a normal state. The normal state is a pristine or reference condition. In every geographic area, biologists find what they call a reference condition or a pristine area, and they look at the benthic macroinvertebrate community in that pristine area, and every sample they take in watersheds around that pristine area, they compare to it. Rapid bioassessment protocols are used because they're cost effective, and research has shown that they're scientifically valid. Another thing that's very important is with these RBP protocols, you can sample many sites in one season. You basically can have a team of scientists go out and collect samples throughout the season. They collect the samples, they preserve the samples, and then they can actually analyze the samples when the weather is bad. They can do that indoors. In addition, the collection of samples and the identification results are pretty quick. You can do them pretty fast. You get quick results, and you can use those results in management decision. Again, these protocols are published in the literature, and they're widely accepted. Reports are easily translated into management and translated to the management and to the public. And the sampling procedures are environmentally benign. They do not negatively impact the environment. So these RBPs are important. They're widely accepted. As you can see, these advantages, RBPs are used because simply in most Western states, there would be no chance of inventorying the thousands of water bodies that are required to be inventoried by the Clean Water Act. So we can use these RBPs to comply with the Clean Water Act in a time-efficient manner. So we can get a lot of data fast, and we don't have to wait years and years and years to get to these streams, when we can send a team of qualified scientists out to take the samples, and we can just work watershed by watershed and eventually cover a state fairly fast. Now, if we take this down to a lower level, and we take it to the region of the Pacific Northwest, and let's use Idaho for an example. We look at Idaho's rapid bioassessment protocols. In Idaho, we call it the BURP program, the BURP program. Idaho's Beneficial Use Reconnaissance Program, or BURP, was initiated in 1993. It was done to integrate both chemical and biological monitoring data with the physical habitat structure of streams. And this whole protocol was done to meet the biological component that's required by the Clean Water Act that was passed by Congress. So if we take a look in a little bit more detail at Idaho's BURP program, Idaho's state waters are considered either, you look in a watershed in Idaho, and the State Department of Environmental Quality will classify a stream as either meeting the designated beneficial uses or not meeting designated beneficial uses. And in Idaho, we have 5 potential designated beneficial uses for surface bodies of water in our state. All bodies of water in the state have at least one beneficial use. All surface streams have at least one beneficial use. And I just wanted to list the 5 beneficial uses that a stream can be classified as having. First of all, a designated beneficial use. It may be for aquatic life support. It may be for the fishery. It may be for primary and secondary contact recreation. A beneficial use may be using that water body for swimming and boating, or boating. Another designated beneficial use of that water body might be water supply. We use that water for irrigation or drinking. Another potential designated use may be wildlife habitat. So we use the BURP program. We do the rapid biological assessment using benthic macroinvertebrates in the watershed. And the score that we get using benthic macroinvertebrates will tell us whether or not the designated beneficial use of the stream is met. If any of the above designated beneficial uses are not met for a particular stream, let's say we have a stream that is aesthetics and water supply. If we do not meet the designated beneficial use, or we don't have enough information to determine if the stream does meet the designated beneficial use, the stream is placed on what's called the 303D list, and further study and/or remediation needs to be done on the watershed. So a western state, or all states in particular, in particular a western state like Idaho, we have all these watersheds, all these surface bodies of water. In theory, all of these surface bodies of water are supposed to be assessed as to whether they meet their designated beneficial use. The BURP program is used to do this, and benthic macroinvertebrates are used primarily to determine whether or not the beneficial use is met. If the beneficial use is met based on the BURP monitoring program, we move on to more problem water bodies. If the beneficial use is not met, it's put on the state's 303D list, and then further study and/or remediation must be done. So let's take a look at how the benthic data is collected. Basically, we know there are many, many species. The taxa is quite great for benthic macroinvertebrates. All the collected species data is run through a statistical package. We go out in the field, we sample for the macroinvertebrates, they're collected, they're brought into the lab, and at least 300 species, or at least 300 organisms from each sample are identified. We use different metrics in statistical packages to determine if beneficial uses are made. Actually, there's 97 different metrics that we can look at. We actually use nine different metrics to calculate what we call a stream macroinvertebrate index score, or an SMI. And we'll take a look at these 9 different metrics in just a few minutes. So what we're trying to do is we're trying to come up with a stream macroinvertebrate index score. And based on that number, we can determine whether or not the stream is meeting the designated beneficial use. The SMI score is an overall score of the water quality based on biological assessment. We use 9 metrics to calculate this score, and we'll show you how it's done in just a few moments. Within the 9 metrics, we're interested in several things. We're interested in how rich the population is of the benthic macroinvertebrates. Do we have lots of benthic macroinvertebrates, or do we have few? We're interested in the population composition, the different taxa of macroinvertebrates that are present. We're interested in pollution tolerance. We know that certain macroinvertebrates are intolerant of certain things like pesticides, or maybe nitrates, or phosphates, or maybe temperature. We're looking at population diversity of macroinvertebrates. The more diversity, probably the better quality of water in the stream. We look at the feeding group, because macroinvertebrates are also food for fish and other organisms. And then we look at habitat. So these things are factored in to the 9 metrics. So let's take a look at the 9 metrics in a little bit more detail. The first metric we're going to take a look at is total taxa. It's really a richness measure that's associated with high taxa richness means a healthier stream. A high amount of total taxa suggests a high amount of niche space, a high quality habitat, and a good food source. So of the 9 metrics, the first one is the total taxa that are present in the benthic macroinvertebrate population. In a table form, the metric is total taxa. The type of measure is how rich is the biological or the benthic macroinvertebrate community. The definition of total taxa is the total number of taxa in the assemblage or in the sample that is taken. And the predicted response to human impact, total taxa decrease with greater human impact. Then we have certain types of species that we're really interested in. We're interested in the EPT species, the Ephemeroptera, I can't pronounce these very well, the Plectroptera, and the Trichoptera. We know them by common names, which we'll look at in just a few minutes. These are basically a richness measure that focus on specific families of macroinvertebrates that are known to have pollutant-intolerant taxa. So we know some taxonomic groups within these 3 categories that are not tolerant to pollutants. So we look at the richness of these taxa. High EPT values are associated with a healthy stream. The more taxa that are present, the healthier the stream is. So if we take a look at some of these taxa, let's take a look at their common names. Mayflies. We're interested in the mayflies. This is what a mayfly looks like. Mayfly is a source of food. There's lots of mayfly taxa as benthic macroinvertebrates. Let's take a look. This is the metric name. We're looking for the richness, or the number of different taxa that are present that are mayflies. And we know that the predicted response is that if humans have had an impact, and major impact, the predicted response is we have a decrease in mayfly taxa. The third measure of richness, we look at the stoneflies. Picture of a stonefly, and it's a lot like the mayfly taxa. We're interested in the richness. How many different taxa of stoneflies are there? The predicted response to human impact is the more human impact on the environment, the fewer stonefly taxa we're going to find. So here's 3 of our 9 metrics right off the bat. We also have another richness taxa. That's our caddisfly. And just like the mayflies and the stoneflies, just like the mayflies and the stoneflies, we're interested in the richness. How many different caddisfly taxa are there? And the predicted response is the more human disturbance there is in the system, the fewer caddisfly taxa there will be. So that gives us 4 of the 9 metrics that we look at. Then we look at the stoneflies again. We look at the percent Plecoptera. It's a composition measure. We know that this particular group of organisms, the stonefly, the nymphs in particular, they require clean, cold, and well oxygenated water for them to have a high composition. So that's our 5th metric. We're interested in the composition of the benthic macroinvertebrate assemblage that are stoneflies. The percent composition of stonefly nymphs. Basically, the more negative impact, or the more impact humans have had on the stream, the fewer or the lower the composition of stonefly nymphs. Then we look at the HBI, the Hilsenhoff Biotic Index, HBI. It's a pollution tolerance measure. This pollution tolerance measure is a weighted average that characterizes the assemblage's overall tolerance to pollution. So again, it's a weighted average that characterizes the assemblage's overall tolerance to pollution. We go to the table, the HBI. It's a measure of pollution tolerance. The definition is tolerance of organisms to pollution. And basically, this index increases with more human impact. The next of the 9 metrics is the percent 5 dominant taxa. This is a diversity measure. We have many different taxa of benthic macroinvertebrates in the stream. We're looking at the percentage of the macroinvertebrates that fall into the 5 dominant taxa. The higher the percentage of benthic macroinvertebrates that fall into the 5 dominant taxa, the less diverse the macroinvertebrates are, and thus the poorer the water quality. So the percent 5 dominant taxa, it's a type of measure, it's a diversity measure, and it's the composition of the 5 most abundant taxa. With more human disturbance, these 5 dominant taxa really dominate the scene, and there's less of the other types of benthic macroinvertebrates present. Then we have 2 more of the 9 metrics. We look at scrapers. Scraper taxa are a specialized feeding group that are considered to be more sensitive to disturbance. So we have scraper taxa functioning. It's a functioning feeding group. The number of taxa that scrape are important. The more impact by humans, you would expect to see a decrease in the number of scraper taxa present. And then finally, we have Klinger taxa. And Klinger taxa indirectly measures the amount of fixed retreats and surface area to attach to by counting the number of Klinger taxa that happen to be present. And this is a measurement of habitat. The tolerance of organisms to pollution, you would expect as there's more human response, you would have a decrease in the number of Klinger taxa. So these are the 9 metrics. There's a tremendous amount of research that went in to developing these metrics, but these metrics are widely used in North America, in Europe, and in many other areas of the world as databases get better. So there's hundreds and hundreds and hundreds of papers written putting these metrics together. So let's calculate an SMI. There are specific formulas that we use in Idaho. Idaho is not all the same. We have a mountain region, we have a prairie region, we have a desert region. There are specific bioregions in Idaho. And in the example today, I'm going to use the mountain bioregion as an example. So we have a formula. Let's take a look at the mountain regions in Idaho for our main metrics. Our total taxa metric, the formula is 100 times the total number of taxa we found in our samples divided by 39. The EPT taxa, the formula is simply 100 times the number of taxa we find divided by 13, 10, or 10. Then the percent plectroptera, the formula is 100 times the taxa divided by 40. Now, if we were in a prairie bioregion, these divider numbers would be a little bit different because the reference stream in a prairie would be different than the reference stream in the mountains or a reference stream in the desert. So the formulas are a little bit different. The formulas differ by the number we divide by. For the other 4 metrics, the HBI score is 100 times 10 minus the HBI divided by 8.4 in the mountains. The percent 5 dominant taxa, calculated this way, the scraper taxa is 100 divided by the number of scraper taxa found divided by 8. The Klinger taxa, 100 times the number of Klinger taxa found divided by 23. Again, the difference between the mountain bioregion and the prairie bioregion and the desert bioregion would be the division numbers right here. We put this all together and we get an SMI score. For the mountain bioregion, we get an SMI score that looks something like this. The score will be someplace between 0 and 100. And based on statistics and previous research, we come up with an ecological health rating. And it can be very good, good, fair, poor, or very poor. And our SMI scores, which are calculated from the 9 metrics, if the SMI score ranges between 84 and 100, we have a very good ecological health rating. If it's 65 to 83, we have a good ecological health rating. If it's between 44 and 64, we have a fair ecological health rating. Between 22 and 43, a poor ecological health rating. And between 0 and 21, a very poor ecological health rating. As you would suspect, that streams that have SMI scores, watershed streams that have SMI scores, between 65 and 100 are good or very good, and they probably meet the designated beneficial use of the water body. Streams that are rated as poor or very poor probably do not meet the designated beneficial use. And thus, more research is required and or remediation, and fair is kind of in between. It depends on the circumstance and what is the specific beneficial use. But basically, we use benthic macroinvertebrates to come up with an SMI score. And the SMI score will tell us the ecological health or the water quality of the water body we're interested in. So a lot of research, a lot of species identification, and it boils down to a simple number between 0 and 100. It's a very useful scale. It's something we can go out and assess a stream in a short period of time. And if we use accepted sampling protocol and we're good at species identification in the laboratory, we can get an awful lot of information that's very valuable. I'll show you the use of a benthic macroinvertebrate index in the next lecture. Thank you for your attention. Designated use might be aesthetics. So we have 5 different designated beneficial uses in Idaho. All streams have at least one designated beneficial use. Several water bodies have more than one designated beneficial use. Okay, so each stream watershed has one or more designated beneficial uses.